breaking the wall of T-cell fitness. Amani Gillette. Falling Walls Lab, Wisconsin.
Imagine that you or someone you love has just been diagnosed with a blood cancer. You're going to take a little bit of time to kind of come to process this, of course, and then you're going to realize that you've been hearing about this novel treatment that takes cells and uses them to treat your cancer, and you're suddenly very excited. This is CAR T-cell therapy. CAR T-cell therapy produces lasting remission in some cancer patients, and it's mostly been driven, the excitement, by stories like the case of Doug Olson, one of the first CAR T-cell therapy recipients who more than a decade later has remained cancer-free. CAR T-cell therapy relies on taking T-cells, a type of immune cells that exist in your body, to fight off infections from cancer patients, and then genetically engineering those cells to fight that patient's cancer. And as I'm sure you can imagine, with collecting cells from already sick patients, sometimes you just don't have the best quality cells to even start this process with. And the sad reality is that over 50% of patients who receive this treatment actually do relapse. And critically, each treatment costs around $350,000 in the US, and that's crazy knowing the US healthcare system, maybe not insane. But that cost is even about 300,000 euros in EU countries where this treatment is offered. So it's just expensive across the board. And this cost is primarily due to the patient-specific nature and the technically and logistically complex manufacturing process, which can take weeks. So the question becomes, how do we break the wall of measuring T-cell fitness before manufacturing so that we can ensure that more patients who get this terrible diagnosis end up with a success story like Doug's? Our solution is based on the inherent fluorescence that is present in all cells. In fact, if I were to shine a laser on my skin at the right wavelength of light, I would be able to measure the fluorescence of my cells, and this is true for all of the cells within a body. Obviously, you need some fancy detectors and fancy lasers, but it does work. And this is important because it means the specific fluorescence that we care about comes from a metabolic coenzyme called NADPH. And this is present in every cell. And crucially for us, it does play an important role in cell fitness. And this is important because it means that the signal changes with any form of metabolic shift, like the one that occurs in T cells when they are quiescent and ready to be turned into cancer-killing CAR T cells. And crucially, our label-free flow system can measure this in real time with up to 92% predictive accuracy of T cells that are fit or unfit for CAR manufacturing. So we believe that our system can help identify cell fitness before manufacturing to help inform whether a patient's T cells will successfully become CAR T cells and ensure that everyone gets a story like Doug's.